In today's topic, we would be discussing microorganisms around us and the concept of culturing. Microorganisms, otherwise called microbes or germs, are living things that cannot be seen with the unaided eye but can be observed using microscopes. They exist almost everywhere, in water, air, soil, on surfaces, and within living organisms. They are carried by air currents from the Earth's surface to the upper atmosphere and occur most abundantly where there is food, moisture, and an adequate temperature for their growth. It was the invention of the microscope that opened the gateway to the world of these minute living organisms. The first person to discover microbes was a Dutch scientist named Antony van Leeuwenhoek, 1632 to 1723. Using a simple microscope, he was astonished to discover that rainwater collected from pools was full of tiny organisms. Groups of microorganisms. Microorganisms include all viruses, bacteria, and protists. Others include cyanobacteria, certain fungi, and algae. Bacteria, these are minute unicellular organisms or simple associations of similar cells, which multiply by binary fission. Most bacterial cells range between 0.2 and 2 in diameter and 0.0005 mm to 0.002 mm in length. Each bacterial cell has a cell wall with cytoplasm but lacks a well-defined nucleus. Consequently, they are prokaryotic organisms. Bacteria exhibit a range of shapes. Some bacteria have long, thread-like structures called flagella, which assist in locomotion. Bacteria with a spherical shape are referred to as cocci, singular, coccus. There are several forms. Streptococci, arranged in chains, they cause sore throat. Cephalococcus stick together to form irregular bunches and cause boils. Diplococci occur in pairs. For example, pneumococcus cause pneumonia. Bacilli, rod-shaped, they cause typhoid fever. Spirilla, singular, spirillum rod-shaped bacteria twisted into a spiral shape. Spirochetes, spiral but more flexible and slender, with a helically coiled structure. An example is Treponema pallidum, which causes syphilis. Vibrio, comma-shaped bacteria. For example, Vibrio cholerae causes cholera. Viruses are a large group of pathogens whose presence is felt only when they are in contact with living cells. They are very small, ranging between 0.1 m and 0.25 m in diameter. The largest virus is less than 1/4 the size of a typhoid bacterium. A virus consists of nuclear material, either DNA or RNA, enclosed within a protein coat. Outside living organisms, they resemble complex chemicals. Protists, these are single-celled organisms, most of which are only visible with a microscope. They are common in fresh water and moist soils. Examples include euglena, paramecium, trypanosome, and plasmodium. Fungi are diverse in form. The blue and green growth on oranges, lemons, cheese, and the white-grey growth on bread are usually signs of fungal infections. Fungi feed saprophytically. Examples include mucro, rhizopus, penicillium, and aspergillus. Algae, most algae are unicellular and very small. They have chlorophyll and occur abundantly in water, moist soils, bark of trees, and stones. Free-floating microscopic algae are referred to as phytoplankton and form the major food source for aquatic animals. Examples of unicellular algae include Chlamydomonas and Protocarchus. Concept of culturing. A prerequisite for studying microbes is their cultivation under laboratory conditions. Hence, it is important to understand the nutrients and physical conditions needed by these organisms. Bacteria, fungi, and algae are typically grown in appropriate media. The material on or in which microbes grow in the laboratory is called a culture medium. Some media are prepared from complex extracts of plant or animal tissues. A culture is the population of organisms cultivated in a medium. If a culture contains only one species of organism, regardless of the number of individuals, it is called a pure or ascetic culture. A culture containing two or more species growing together is called a mixed culture. An important medium used for growing microbes is agar. It is a dried polysaccharide extract of red algae used as a solidifying agent. Agar is not broken down by microbes. Identification of microorganisms. There are many methods for identifying microorganisms around us. These include the use of microscopes, stains of different types, types of colonies formed by the microbes, their food requirements, and their oxygen requirements. To determine the presence of microbes around us, suitable media are used to culture them in petri dishes, which are first sterilized by heating them in a pressure cooker, autoclave, or oven. Microorganisms are not capable of growing in the air. The exposure of nutrient agar to the air will show the growth of different bacterial colonies. Microbes commonly found in the air include viruses, bacteria, and fungi. Microbes in aquatic habitats may be grouped into natural water, soil, and sewage microbes. Examples of the first category include protists, algae, and some fungi and bacteria. Examples of the second group include rhizobia, nitrosamines, and nitrobacteria. Examples of sewage microbes include Entamoeba histolytica and Escherichia coli. Microbes living in our bodies form a normal population without causing harm. However, under certain conditions, they may become dangerous. Pathogenic organisms cause diseases when body resistance is low or when normal microflora are disrupted by the use of antibiotics. Any food item left unpreserved for a long time will spoil due to the activities of microbes. Pathogens enter the body through four main routes: air, food and water, contact, direct or indirect, and insect bites or cuts. Carriers of microorganisms. Any agent that carries microbes from one place to another is called a carrier. Carriers can be living or non-living things. Non-living carriers include air, water, and food, while animals, e.g., houseflies, mosquitoes, rats, cats, are living carriers. Animal vectors carry pathogens either mechanically or biologically. In the mechanical method, animals carry the pathogens on their bodies, where they cannot grow or multiply. In the biological method, the vector becomes infected by feeding on the body fluids of infected persons or animals. Thanks for watching. See you in the next video.